Hello everyone, this is JJ here, and today we will meet cells. I'm so excited. I hope you are also. Well, I am excited because I believe they are the most beautiful, tiniest thing in the whole universe. And do you know why? Yes, because they make us. And you already know that we have so many different kinds of cells in our body, few that kill monsters, well, by monsters, I mean foreign particles, and few that store our fat, and few that store our good memories. That is, they take those memories to our brain and store there, right? So, yes, we have grown so big, of course, so we need more cells to fill all the spaces and also to be alive. So, yes, cells also need to have babies. By babies, I mean they also need to undergo division and multiply in number, but this should happen in a very controlled manner. And if there is no control, then this happens. And when this happens, there is no space and no food. And this is because of the lack of apoptosis. And if you don't know what is apoptosis, then don't worry, you can click on the card above and know more. So, yes, back to the topic. So, if this happens, the cell dies eventually, which of course we don't want because it's sad. So, for that, we have a control department within our body, and I am not joking, right? So, before that, we should know all different phases of the cell. So, here we have interphase, which is pretty long, and it's a phase where the cell really does so much hard work for being capable of dividing, just like how we work hard for cracking exams, right? And then there is a tiny mitotic phase where finally the cell divides. So the interphase is divided into three different phases, that is G1, S and G2 phase. Okay, so the G1 phase is the phase where the cell really grows very big, makes organelles and stores all the protein it might need for the survival in the next phase, that is S phase. And the cell stays in this phase for almost 11 hours, which is crazy and pretty long. And then the cell is given two choices, that is, it can either proceed to work hard in S phase, or it can take rest in G0 phase or G0 phase or resting phase. Okay, because at times we don't need so many cells, so few of them can take rest and divide later. Yeah, or sometimes some cells do not divide at all, that is, just like our neurons. Neurons are the best examples for this. They do not divide after they are formed. Okay, so they are permanently in G0 phase. So yes, the next phase is 8 hours long, and here some action happens, and that is DNA replication occurs. So if when someone asks you, and that is in which phase DNA gets replicated, you already have the answer, that is S phase. Now in the G2 phase, again proteins are synthesized, but this time for final division. Okay, so what type of proteins? The proteins such as tubulins are synthesized here. Such as these proteins, that is tubulins, are involved in the formation of microtubules or the spindle fibers, right? And the spindle fibers are attached to chromatids, and it is needed in the mitotic phase. So we should be ready for the mitotic phase, right? So in G2 phase, all such proteins are synthesized and stored. And finally, we have mitotic phase where the cell divides. So here we have prophase, prophase, metaphase, anaphase, and telophase. And all this takes just single hour, that is just 1 hour, which is pretty cool. So remember, while you're listening to this video or watching this video, your cells might be really working hard and dividing. But wait, they cannot just move around easily. There are checkpoints where the system checks whether they are cheating or not. And if they are found guilty, then they are sent out of the cycle and not out of the class. Yes, so the first checkpoint is start checkpoint. Well, there are three major checkpoints. There might be so many other checkpoints, but here we'll discuss three major checkpoints. Okay, so checkpoint 1, which is known as start in yeast and restriction point in vertebrates. Okay, and this is present in late G1 phase. That is, if this is G1 phase, it is present somewhere here, which means it is present in late G1 phase. And they check whether the proteins are present for replication or if some organelles are missing, etc., etc. Which simply means it checks whether the condition is favorable for entering into next phase or not. Okay, and then comes the most important one, that is G2M phase. That is, it is present in between G2 phase and M phase. Okay, and it checks whether the replication is completed. That is, it occurs after S phase and G2 phase, right? So it has to check whether replication has completed and assures that there is no damage, and then gives the green signal, yes. And then it finally enters into the mitotic phase, right? And then the last checkpoint is metaphase to anaphase transition checkpoint, and this checks whether the spindle fibers is properly attached or not, just like checking whether your answer sheets are properly tied or not. If it's not proper, then you lose your marks, and here the cell loses some very important genetic information which could be really, really dangerous. Yeah. And now we'll meet the HOD of cell cycle controlled department. So, which is cyclin and cyclin dependent kinase. So, this is cyclin and cyclin dependent kinase. So, they both are friends and they work together. While cyclin regulates its friend CDK or cyclin dependent kinase, who is a protein kinase enzyme. So this is a kinase enzyme. Kinase is for enzyme is for phosphorylating. Okay, they phosphorylate other proteins. So this CDK phosphorylate the target proteins when they come together with the cyclin. Okay, so cyclin and CDK come together and they phosphorylate serine and threonine residues or specific target proteins which is involved or which is needed to move on to the next phase. Okay, so these proteins is important for the cells to move on to the next phase. Yeah, so the CDK is not always active. For making CDK active, we have to do some imagination. So imagine CDK has a protein slab on its head which is really heavy, and because of which it cannot just work. So cyclin, which is lightweighted, comes and displaces this protein slab and makes CDK partially active. Remember, it's partially active and not completely active. So for making it completely active, there is another kinase which is CDK activating kinase or CAK, simply CAK. So it is also a kinase, which means it also phosphorylates, right? So this time, this CDK activating kinase phosphorylates the threonine of CDK itself. Okay, so this CDK activating kinase, as the name suggests, it activates CDK, right? It activates CDK by phosphorylating the threonine of CDK, and finally it's completely active, right? And now as it is completely active, it can accommodate ATP too, because ATP is needed in CDK's functioning. Yeah, so now for each assignments in these phases we have paired cyclin and CDK complex, so we will learn that right now. So these are the four cyclin CDK complex, and they of course help in transition from one phase to another phase, right? So the first one is G1 CDK for G phase, and the second one is G1S for transition from G1 to S phase, and then we have SCDK for S phase and MCDK for M phase. There is no G2 M phase or G2 phase. And then we have four type of cyclins, that is cyclin D, cyclin E, cyclin A and cyclin B. There is no cyclin C. Okay, you can learn the word deep maybe because it might help you. Yeah, remember there is no C and it starts from D and ends with B. Okay, so it is deep, cyclin deep. Yeah, and then we have their CDK partners. So the first one has two CDK, right? it has CDK4 and CDK6, whereas the second and the third has the same CDK, that is CDK2, and the last one is CDK1. So we'll just revise once. Okay, so the first phase has cyclin D and CDK4 and CDK6. So cyclin D and CDK4 or 6 comes together and forms this complex, that is G1-CDK complex. Yeah. Similarly, cyclin E and CDK2 comes together and forms G1-S-CDK. Yeah, and the cycle goes on. You have to learn the cycline's name because it is really important. So this is DEAB, and then that is deep. Okay, and then in the case of CDK partners, we have two CDKs for the first one, that is G1 phase, that is 4 and 6, and we have two similar CDK for the second and the third one, that is CDK2, CDK2, and then CDK1, and then that's simply over, right? So we have learned that. Yippee. Okay, you can tap your back right now. You should be proud of yourselves. And then now we will learn the next thing, that is there are ways through which CDK can be inhibited, which is necessary too, because just one type of CDK is not supposed to do the whole job, as we already have four different types of different phases. So according to different phases in which they are in, the others or the not working ones are inhibited or activated by different kinds of enzymes. And the first enzyme is V1, which is a protein kinase. A kinase, which means it phosphorylates, right? So this particular enzyme phosphorylates the tyrosine of at position 14 of the CDK, right? It phosphorylates the tyrosine of CDK at position 14 when they are not in use. Similarly, when they are needed, this particular tyrosine, which was phosphorylated by V1, is dephosphorylated by phosphatase enzyme CDC25. Phosphatase means it removes phosphate group, right? So CDC25 does this job. It removes the phosphate group and hence activates that particular CDKs. Yes, tada, and that's all, right? And we have another set of enzymes which is known as CDK inhibitor proteins, or just CKIs. Okay, CDK inhibitor proteins or CKI. They are of two different types, that is CYP or KIP kinase or INK4 kinase. Well, CYP stands for CDK interactin protein. Okay, CDK, I for interactin and B for protein, and KIP stands for kinase interacting protein. Okay, they both are kinase, which means they also add the phosphate group, and they can bind to every cyclin CDK complex. Okay, they have the superpower. They're the super powerful villains which can bind to every cyclin CDK complex and inhibit their activity. So it includes P21, P27, and P57. Okay, we have P21, P27, and P57 in CYP and KIP kinase. And the next one is INK4, which stands for inhibitors of CDK4. And do you remember in which phase CDK4 was included? Well, you can pause this video and think for a second. Where was CDK included? That is, in which phase was CDK4 included? Yeah, the answer is G1 phase. The first phase with two CDKs, that is 4 and 6. Well, it's inhibited when INK4, okay, this particular enzymes comes in between the two friends, that is cyclin D and CDK4 and CDK6 of G1 phase. So you know, right, cyclin D is present in G1 phase, and they have four teammates, that is P16, P15, P18, and P19, which is also known as INK4A, INK4B, INK4C, and INK4D. That's all about the inhibition and activation of cyclins and CDK complex. So actually, if we go deep, we have two more inhibitory proteases, that is ubiquitous proteases APC and SCF complex, which I will be able to explain you in a better manner only if I tell you about the phases and its regulation, which I'll be discussing in the next lecture. So until that, you have to wait. It will be provided soon to you. So yes, thank you for bearing with me until now. Please don't forget to share and subscribe. It encourages me. And yes, much love. Bye from JJ.